acid-base balance and discussing the role of kidneys, the third line of defense. We have previously discussed the role of chemical buffers and the respiratory system as the first and second line of defense in acid-base balance. So the kidneys, they are the ultimate responsible for the correction of acid-base imbalance and feedback gain of 100% for a healthy kidney. The kidneys can respond to acid-base balance by these three mechanisms: by secreting hydrogen ions, reabsorbing filtered bicarbonate, and production of new bicarbonate ions. The kidneys handle bicarbonate like it is filtered. We can calculate this by plasma concentration of bicarbonate multiplied by the GFR, and this is the filtered load, on average 4,320 milliequivalents per day. 85% of this bicarbonate is being reabsorbed on daily basis from the proximal convoluted tubule, 10% from the thick ascending limb of the loop of Henle, and only this 5% is subjected to variation in the collecting tubules, which may be increased or decreased, reabsorbed or maybe excreted more, reabsorbed less, depending upon the acid-base status of the body. This is how the kidneys can respond to acidosis, by retaining all this 5%, or alkalosis by excreting this 5% or as much as required. Renal handling of hydrogen ions. The renal tubular cells, when reabsorb hydrogen ions, alternatively they can secrete hydrogen ions while reabsorbing bicarbonate ions. This is a proximal convoluted tubular cell which is showing the reabsorption of bicarbonate and secretion of hydrogen ions by the carbonic-anhydrase mechanism. What happens? The carbon dioxide combines with water to form carbonic acid, which splits into hydrogen and the bicarbonate ions. This bicarbonate ion is then reabsorbed from the basolateral border by the sodium bicarbonate co-transport mechanism, as you can appreciate over here. Whereas the hydrogen ion that was produced is secreted out into the renal tubular lumen by, again, a counter transporter, sodium hydrogen counter transporter. This hydrogen ions, when released in the renal tubule, cannot stay like this because it will drastically decrease the urinary pH very quickly. To inhibit this from decreasing the urinary pH, it has to combine with the urinary buffer, that in this example is bicarbonate buffer. This bicarbonate is the filtered bicarbonate. This is combining with this secreted hydrogen ion, forming the carbonic acid again, splitting into carbon dioxide and water. And this carbon dioxide is then reabsorbed and secreted via the lungs or combined with water to form the carbonic acid. So ultimate resultant of all these steps is the reabsorption of a filtered bicarbonate. This bicarbonate was filtered, not directly reabsorbed, but reconstituted and reabsorbed, but the resultant is the same filtered bicarbonate is indirectly reabsorbed. Plus, in return, one hydrogen ion is excreted, as you can see. This is occurring in the proximal convoluted tubule. And another thing you can appreciate, that hydrogen ion is being secreted by the secondary active transport, whereas in the collecting tubules, where the hydrogen secretion and bicarbonate reabsorption occurs, this is almost following the same pathway. The carbonic acid forms under the influence of carbonic-anhydrase, splits into hydrogen ions and bicarbonate. This bicarbonate is being reabsorbed. The channel, however, is different. Chloride bicarbonate exchanger over here. Plus, other thing you can see, hydrogen being secreted is using a primary active transporter, hydrogen ATPase or hydrogen potassium ATPase, and this is occurring by the type A intercalated cells, which reabsorb potassium and secrete the hydrogen ions. By this primary active transporter, this has a capacity to secrete much more hydrogen ions as compared to the secondary active transporter in the proximal convoluted tubule. So, on daily basis, the hydrogen ions being produced in the body are being secreted from the renal tubules. Those being secreted are buffered with the bicarbonate ions. That being buffered with the bicarbonate in the urine, we call it the volatile acid being secreted. This is approximately 4,320 milliequivalents per day, which is equal to the bicarbonate being filtered every day. So for each hydrogen ion being secreted, there is one bicarbonate ion being reabsorbed. In comparison, the acid which is being secreted in excess of this bicarbonate being reabsorbed, we call it non-volatile acid load, and this is approximately 80 milliequivalents per day. This hydrogen ion being secreted in the urine is then buffered with other buffer systems other than the bicarbonate, for example, phosphate-buffer and the ammonia-buffer. So total of non-volatile plus volatile acid is approximately 4,400 milliequivalents per day of acid is being secreted. This is the summary of what is happening in the PCT and early DCT. The hydrogen ions being secreted by the secondary active transport. In return, one bicarbonate being absorbed and this hydrogen ion being buffered with the bicarbonate ions. The minimum pH that is achieved by this action is in urine 6.7 and 95% of bicarbonate is being absorbed, 85% in the PCT and 10% in the thick ascending limb and early distal convoluted tubule. 95% absorption being occurring by this way, 95% hydrogen ions being secreted by this way. Still the pH of urine has only decreased to 6.7. Whereas in the late distal convoluted tubule and collecting tubules, where the hydrogen secretion is occurring by the primary active transport, here the hydrogen ion secretion and bicarbonate absorption is 5%. And these hydrogen ions are being buffered with the phosphate and ammonia buffers, and the urine pH minimum that can be achieved in these segments is 4.5. So normally the hydrogen ions secreted per day are 4,400, whereas bicarbonate reabsorbed is 4,320. The difference 80 milliequivalents per day on average healthy adult is being secreted as non-volatile acid titrated with the phosphate or the ammonia. Whereas in case of acidosis, the hydrogen ions will be much in excess as compared to the bicarbonate ions. Bicarbonate will be completely reabsorbed and the rest of the hydrogen ions will be excreted in combination with phosphate and ammonia. If the patient is suffering from alkalosis, there would be less bicarbonate reabsorption. Excessive bicarbonate ions over hydrogen ions, those in excess of hydrogen ions will not be reabsorbed, rather secreted in the urine to correct for the alkalosis. Now the third mechanism of kidney to respond to the acid-base imbalance. First two we have seen, the reabsorption of bicarbonate and secretion of hydrogen ions. Now comes the third one, which is generation of a new bicarbonate ion, and that occurs while secreting the non-volatile acid. Here you can see the rest of the mechanism is same. Carbon dioxide and water combining to form carbonic acid, bicarbonate being reabsorbed by simple diffusion, the hydrogen ion being secreted. This is by counter transport, and while reaching the tubular lumen, it is combining with a non-bicarbonate buffer, in this case sodium phosphate, making the phosphoric acid, and this hydrogen becomes trapped with this acid, has a minimal impact on the urinary pH and cannot diffuse back to the renal tubular cell, and the bicarbonate is absorbed. So the net impact is addition of a new bicarbonate ion to the plasma. This is a bicarbonate ion which was not already filtered but was generated and reabsorbed. So this is the third mechanism of the kidney to respond to the acidosis. Similarly, a new bicarbonate ion is added when hydrogen ions combine with the ammonia buffer in the tubular lumen. As you can see, the rest of the process is same. Bicarbonate ion produced and reabsorbed in the plasma, whereas hydrogen ion, it combined with the ammonia to form the ammonium ion. This hydrogen ion got trapped, secreted by the active transport, cannot diffuse back to the renal tubular cell, stays in the urine, contributes to the urinary pH, and in return, one new bicarbonate is added in the blood to combat the acidosis. From where is this ammonia coming? This is coming from the metabolism of glutamine in the renal tubular cell, which breaks down to form two bicarbonate and two ammonium ions, which combine with the hydrogen ions to buffer for the urinary acidity. In case of chronic acidosis, the metabolism of glutamine may be increased to provide more ammonia buffer constituents. So kidneys correct the acidosis by completely reabsorbing bicarbonate, adding new bicarbonate through the formation of ammonium and titrable acid that corresponds to the phosphate buffers, removal of excess hydrogen ions in combination with urinary buffers like ammonium and the phosphate buffers. In chronic acidosis, the production of ammonium will also be increased. Kidneys correct for alkalosis by incomplete reabsorption of bicarbonate as it incompletely titrates with the hydrogen ions. Increased renal excretion of bicarbonate means addition of a hydrogen ion to the ECF, which will correct for the alkalosis. In addition, there are other modulating factors which can modulate the reabsorption of secretion of these hydrogen and bicarbonate ions, including increased partial pressure of carbon dioxide. When this is more, there is going to be more formation of carbonic acid and hydrogen ions. So hydrogen ion secretion is going to increase. That means alternatively bicarbonate reabsorption will increase. In presence of increased angiotensin II, this directly stimulates sodium hydrogen exchanger and sodium bicarbonate exchanger, thus increasing the secretion of hydrogen ions and reabsorption of bicarbonate ions. Sometimes in patient of dehydration, there is excessive production of angiotensin II, which secondarily stimulates the sodium hydrogen and bicarbonate transporters, resulting in alkalosis, and we call it contraction alkalosis. Angiotensin primarily was produced to combat the dehydration but also resulted in alkalosis. Decreased aldosterone level. As we know, aldosterone is involved with the secretion of potassium and hydrogen ions. When its level is low, hydrogen secretion will decrease and bicarbonate reabsorption will also decrease. Decreased ECF volume. When the extracellular fluid volume will decrease, overall reabsorption of substances in the proximal convoluted tubule will increase, also resulting in increased reabsorption of the bicarbonate ions and secretion of hydrogen ions. Hyperkalemia will stimulate the production of aldosterone. Also, potassium and hydrogen ions, they compete for each other. When the person is suffering from hyperkalemia, the intercalated cells type 2 will be hyperactivated to secrete the excessive hydrogen ions, and in return the hydrogen ion secretion will be decreased as they are competitors. Also, the reabsorption of bicarbonate ions will be decreased, and the mechanism is its section on the PCT as well as the collecting tubules. This is the summary of the factors which can increase hydrogen ion secretions. For example, acidosis, carbon dioxide is more, hydrogen is more. Decreased extracellular fluid volume will increase the bicarbonate reabsorption and increase the hydrogen ion secretion, as overall exchange across the PCT will increase. Angiotensin II by stimulating sodium hydrogen and sodium bicarbonate exchanger. Aldosterone directly stimulating the sodium, potassium, hydrogen and bicarbonate exchange. And hypokalemia. In hypokalemia, intercalated cells type A will be more activated and potassium will have to be retained. When potassium will have to be retained, of course, in return, hydrogen will have to be excreted. Similarly, diuretics also have impact on the acid-base balance. The diuretics other than carbonic-anhydrase inhibitors are involved in causing metabolic alkalosis mostly, but the carbonic-anhydrase inhibitors decreases the absorption of bicarbonate and may result in acidosis. And that's all about the renal regulation of acid-base balance.